Okay, so today I will tell you about a current project I'm working on in the Anton Goluborotko groups in collaboration with Daniel Gerlich at IMBA in Vienna. And okay, so as Chris, sorry, I cannot, okay. So understanding how the genome is organized within the nucleus and which are the mechanisms that can lead to such an organization and how this three-dimensional organization is linked and coordinated to essential cellular function, it always been a great challenge for both biologists but also theoretical and experimental physicists. As Chris already introduced, a breakthrough towards a better understanding of the spatial organization of the genome, it has been made in 2009 with the development of the high-throughput sequences chromosome conformation capture technology, or briefly Hi-C. With this technique, we are able to retrieve contact maps for whole genomes within the same chromosomes or between different chromosomes. So here there is an example of a...
Sorry. I'm sorry to interrupt you, but we see the presenter view of the slides. So we don't see your full screen. Maybe it would be better to make the full screen so we can see. If you have more screens, maybe you should share the right screen. I don't know.
Sorry.
I'm not an expert.
Yeah. For simplicity, just play the keynote. Yeah.
Okay, so sorry, I will just remove the screen. It'll be easier.
Zoom also has the feature to share a box, a portion of your screen, if you can't change it.
Okay, now can you see the correct screen?
Perfect, yes.
Okay, sorry, thank you. So on the right, you can see a contact map for the same chromosome or for different chromosomes. So in each pixel, you can see the frequency of contacts between two genomic positions along the same sequence or between different sequences. What was discovered with this Hi-C technology? So we already know that the double-strand DNA is wrapped around histones to form nucleosomes, but on a larger scale, the genomic organization was still very mysterious. So Hi-C allowed us to discover that larger genomic regions are organized into loops that bring into proximity pairs of genomic position, for example, for gene regulation purposes. And if we zoom out, we discovered that groups of these loops are clustered together to form topological associated domains, or TADs. We can see this feature in Hi-C maps represented as dots, so as peaks of enriched contacts for loops, or as enriched contacts square for TADs that are insulated one from another. So a position within a certain TAD will have a higher probability to contact a region within the same TAD than another position outside of its TAD. If we zoom out further, we see how groups of TADs are grouped together to form chromosome compartments. These chromosome compartments are mostly actively transcribed, or inactive, and they are named A and B compartments. And we can see them in Hi-C maps showed as the very characteristic checkerboard pattern. So each square will represent a different compartment that will be transcriptionally active or inactive. And finally, we discovered that at the nuclear scale, chromosomes tend to form territories, so tend to prefer to interact within themselves than between them. But beside this numerous insight, Hi-C techniques have several limitations, and there are also several ambiguities in their interpretation. First of all, they are frequencies of contacts, and they do not represent three-dimensional distances, and they provide only information about pairwise interactions, so higher order interactions cannot be distinguished. And then, as already Chris introduced, they are population average maps. So we cannot know which exact interaction are occurring in which cell. So this makes difficult to disentangle mutually exclusive versus co-occurring interactions, and also to disentangle contact patterns associated to different three-dimensional structures. So contact patterns for loops or for tags that overlap in the average map. Moreover, they are a static snapshot of an average population of cells, so they cannot provide directly dynamic or mechanistic information about the system. So which mechanisms can lead the system to such a conformation? And that's why developing theoretical models is so important. So a theoretical model, for theoretical models, what is usually used is are coarse-grained polymer models that can provide a statistical ensemble of conformation whose statistical properties can be compared with the ones found in experimental cells and retrieve both structural and mechanistic information. So from a structural point of view, we can better understand the contact pattern 3D structural association, and from a mechanistic insight, we can discover the mechanism underlying such an organization. The best supported polymer model, physical model for internal chromosome organization is the loop extrusion model. The main character of the loop extrusion model is cohesin. Cohesin is a ring-shaped protein, part of the SNC complexes family, which has two ATPase domain, which are likely to confer to the protein a motor-like activity, a motor activity. The loop extrusion model says that once a cohesin protein is loaded onto the DNA, it can actively extrude loops through its soul until it encounters either two convergent CTCF sites, here represented with different arrows of different colors, and where it becomes stuck, or when encountering another loop on the right, and it will become stuck for encountering the other loop. Cohesins are dynamically loaded and unloaded onto the DNA, so they basically reshape internal chromosome conformation throughout the cell cycle. This model is the best supported model because it's the only model that can explain most of the features that we see in experimental contact maps. For example, it can explain tags insulation and flames arising from a unidirectional extrusion of loop when encountering a CTCF site only on one side, or for example, corner peak and peak grid that can arise from single loop or nested loops. So on the right, we have a comparison from a simulated map generated with a loop extrusion model and an experimental map within the same genomic region. However, we have I gave an overview of what we know today about chromosome organization into the nucleus. However, a fundamental piece is missing for having a global understanding of chromosome conformation. In fact, chromosome after the synthesis, so after DNA replication, each chromosome sequence is duplicated and to have basically two identical sequences called sister-chromatids that are tethered together until cell entries into mitosis. So we have now a better understanding of internal chromosome conformation, but we don't know how sister-chromatids interact between each other, even if they spend a large part of the cell cycle being tethered together. So we suspect that the final conformation would be a combination of loops within the same sequence and rings topologically entrapping the two different sister chromatids. Since high-C technology, it's a sequence-based technology, it couldn't differentiate the identical sequences of sister chromatids. And so we didn't have until now experimental data about interaction between these two identical sequences. And as a consequence, with no experimental technique, there is also a lack of models of this system. Of sister chromatid cohesion. So the mechanisms of rings entrapment of two sisters is called cohesion. But a few months ago, the scsHi-C technique, sister-chromatid-sensitive technique, was published at IMBA from the Gerlich lab at IMBA. And with this new technology, we are able to discriminate between contacts happening within the same sequence from contacts happening between the two different sequences. So cis contacts within the same sequence and trans contacts between the two sister chromatids. The main finding of this experiment was that while in cis we continue to see TADs and peaks formed by loops, squares and peaks formed by TADs and loops, in trans, so between the two sisters, we see an enrichment of contacts at the TAD boundaries, suggesting that cohesion is happening more often at TAD boundaries. So the two sisters are trapped at specific regions. However, when they depleted loop extrusion mechanisms in cell, they saw a decreasing looping activity, so a decreasing peaks and squares deriving from loops and TADs, and surprisingly, sister chromatids became more paired. And you can see that with the enrichment of contacts along the diagonal of the high-C map. So this is also obvious when we plot the contact probability versus the genomic distance. The contact probability curve is a powerful statistical tool that tells us about the statistical properties of the system in a position-independent way, because high-C maps are position-specific. So we studied contact probability, and indeed, in absence of loop extrusion, so the red curve, the trans-sister contact probability increases for short distances. So comparing red and black curves, indicating an increase in pairing and a decreasing misalignment between two sister chromatids. So this is a sketch of what we understood from this contact probability maps plot. So our main hypothesis is that loop extrusion mechanisms may push cohesive rings to that boundaries, while when we remove loop extrusion mechanisms, the cohesive rings can more uniformly distribute between the two boundaries, and also relaxing the misalignment. A final result from this paper was that actually the heterogeneous we have heterogeneous TADs. It means that there are more pair and less pair TADs, both with an enrichment of contacts at boundaries. And it seems that more pair TADs correlate with transcriptionally inactive domains. So transcriptionally inactive domains are likely to be more paired and less misaligned. But now that we have this new tool, we really need new theoretical models that help us to understand this system. First of all, theoretical models will help us explain the structure behind the contact patterns observed in this new type of experimental data. And then a theoretical model will also complement experimental data, giving mechanistic information that cannot be gained experimentally, so giving insight about dynamical mechanisms, and also make prediction on the impact of sister chromatid organization on essential cellular function, for example, DNA repair or transcription. So what we really want to address with our new the theoretical model that we are trying to develop is which is the distribution of these cohesive rings along the genome, specifically characterizing the systems in terms of average frequency and misalignment. Then we would like to integrate the cohesion model with the loop extrusion model. And then we would like to know if there are other possible mechanisms that can lead to such a distribution of cohesing rings. For example, motor-driven cohesion instead of just diffusion of rings, or transcription correlated cohesion. And how are these structure and mechanisms linked to cellular functions? All this work, it has been done with, I've been doing this work in collaboration with the Gurley Club. So we have preliminary results in terms of understanding how the main parameters of the system, so the average frequency and the misalignment, impact the three-dimensional contact probability in 3 dimensions. Basically, we wanted to understand what happens when we change these parameters in 1 dimension, how it is impact the contact probability in 2 dimensions. And to do that, we model our 2 sister chromatids with 2 linear polymers shown in blue and in red, linked at several genomic positions. This fixed link that you can see in black have an average, a fixed average frequency and a misalignment that can be determined, that is randomly chosen following a Gaussian distribution. And so sigma will be the standard deviation of the misalignment. The misalignment is in average 0. So the genomic separation between I and J will be the same as I prime and J. And we computed the contact probability in 3 dimensions. So we see how changing the frequency, the average frequency between consecutive links doesn't impact the probability of contact within the same sequence, but impact the probability of contact between 2 different sequences. How? It seems that the average frequency between consecutive links determine the interaction point between cis and trans curves. It means that when we pick a position, for example, in yellow on 1 sister chromatid, the probability to contact a genomic position that is closer to the average frequency, so in green, it is higher than to contact the same genomic position on the other sister chromatids, here represented as in purple. Instead, when we change the average misalignment, so when we change the average misalignment, we see how the trans-contact curves start presenting a plateau. And this means that contact probability between position closer than sigma will have the same probability, while contact between positions that are more far apart with respect to sigma will have a smaller probability of contact. So the 2 main rules here are the average frequency determine the point of interaction between cis and trans probability contact, while sigma, so the misalignment, determine the end of the plateau in the trans contact probability. Surprisingly, when we increase sigma in order to have a misalignment that is way greater than the average frequency, we observed that the plateau extended and passed the cis contact frequency. It means that when, in the last picture on the right, when we have a greater average misalignment, the point in yellow will have a higher probability to contact the point in purple on the opposite sister chromatid with respect to contacting the same genomic position in green within the same sequence. So with this analysis, we basically list rules. We list rules that will help us to understand the statistics of the system in experimental results, so in experimental contact probability. And here, for example, we can see that the average, the misalignment, it's a bit less than 10 to the 6 for in absence of loop extrusion, while with loop extrusion is greater than 10 to the 6. And comparing the intersection point, we can also infer the average distance between 2 cohesive rings. So now that we understood the statistical rules that govern the physical system, we would like, and we understood how these parameters actually impact the probability of contact in 3D, we would like to address, and we are addressing right now, when we explicitly models rings on linear chains, how do these rings distribute along the two linear chains if we let them freeze to just diffuse. And then we would like to compare the average frequency found in this simulation with our contact probability. Then we would like to integrate the loop extrusion mechanisms with a simulation of rings threaded on linear chains, and we will see we would like to know how loop extrusion is coordinated and is coordinated with ring diffusion. We would like also to know if with diffusion alone we can reproduce experimental data, or if there are other mechanisms that can replace rings. For example, the pushing by loop extrusion, or for example, transcription activity. And in the long run, we will, once we found a model that can really agree with experimental data, we would like to study and make prediction about how perturbing the system, so perturbing the models, this would impact, for example, DNA repair or transcription activity in experimental data. And this will be done in collaboration with the experimental lab of Daniel Gerlich. So I would like to thank all the Global Collab and the Gerlich Lab, especially Daniel, Sophia, and Michael, who are the people I'm collaborating with, and also my founding sources that will allow me to carry out this project.
Thank you, Flavia. Are there any questions?
Hello, I had a question. So yeah, okay. Can I go ahead?
Please. Yeah.
Yeah. Thanks for the great talk. So I was wondering. So I've been thinking about this for a little bit. So how do you envisage loop extrusion working on trans pieces of chromosomes?
So loop extrusion. You mean loop extrusion in cis?
No, exactly. So loop extrusion works in cis, as far as we know. In the moment that you allow for cohesion of sister chromatids, then you need a mechanism for loop extrusion in trans.
Yeah.
How do you?
So our main hypothesis is that when once cohesion is loaded onto the DNA, it will start extruding loops, and when it will encounter a cohesin that anthropologically entraps the two sister chromatids, it will start pushing this ring until they both reach the CTCF boundary.
Yes, so this is how it works in cis. But so in trans, there is no knowledge of what is outside and what is inside, right? It's not like if in cis there is a knowledge of what is outside, so when you extrude, and what is inside. But in trans, so once that you grab the other piece of chromosome, how do you decide where you go, for instance? Have you thought about that?
So you're assuming that actually cohesive rings, so loop extruding rings, can extrude loops taken in both the sequences.
I don't know. Am I? So what do you think about this? Yes, exactly.
So there is no evidence for that.
Okay.
And so I honestly, I don't know, because there is no experimental evidence for that. So for now, we are assuming that each cohesion ring can extrude only loops coming from the same sequence.
Okay. Okay. And the other, and then just a short comment and saying that if you're looking into basically how sliding rings or slip links organize just polymers, then there is quite a lot of literature in what is so-called entropic competition. And so there is quite a bit of literature on that. So in that direction, you will find a lot of also analytical stuff for to basically work out what is the distribution of slide rings on just fluctuating, of diffusing slide rings on fluctuating polymers. So that is just a comment. Okay, thanks a lot.
Okay, thank you. One more question from Pier Francesco, please.
Yes, hi. Thanks a lot for the talk. Maybe I'm asking a naive question or too far away question, but I didn't know anything about this, and it reminded me a lot of what people did in proteins, where you have like the sequence of amino acids, and you want to predict the 3D structure of the proteins, which amounts to predict the contacts between amino acids, which is what people want to do because the 3D structure of the protein is functional, right?
Mm-hmm.
So there, what they do is that they extract this precisely from the sequence. And I don't know if it's completely crazy to go through DNA and look at if there are sites or portions of the DNA sequence where you can you can show that that are conserved among different, I don't know, types of cells or individuals, and that can help this game of reconstructing the structure.
Yes, so there are some experimental works that shows that maybe cohesing rings can are more likely loaded on certain sequences in the genome than the others. But then I have to say that loop extrusion, but also cohesion, it's a mechanism that happens throughout the cell cycle and along all the genome, and it is highly dynamic. So we think that there are some basic rules that are position independent, so also sequence independent. And probably we will try to find a model to find a model that is absolutely general, so absolutely sequence and position independent, and maybe then we will add some more molecular detail, for example, simulating CTCF sites. CTCF sites have a very specific sequence motif, so maybe we can add this detail into our simulation and see, adding these details, how our model works.
Okay, thank you.